Hello, our project is Emerging Treatments of Cancer, determining what genes have the highest rates of mutation in non-Hodgkin's lymphomas. Determining genes with high rates of mutation can have implications for use in cancer therapies to help the immune system recognize and target the mutations of interest. Results show that the KMT2D, BCL2, and CRE-BBP genes have the highest rate of mutation in certain non-Hodgkin's lymphomas. In this project, various non-Hodgkin's lymphomas were compared to find the highest rate of gene mutation. TP53 and NRAS are genes that code for multifunctional proteins that are critical to cell cycle regulation, apoptosis, cell survival, gene transcription, response to stress, and DNA repair. Lymphatic cancers attack the lymphatic system, which is a part of the body that regulates germ-fighting mechanisms. These types of cancers can attack the lymph nodes, spleen, thymus gland, bone marrow, and many organs. Lymphatic cancers can be categorized into Hodgkin's lymphoma and non-Hodgkin's lymphoma. This project delves into non-Hodgkin's lymphomas, which arise from abnormalities in the body's lymphocytes, also known as white blood cells. More specifically, acute lymphoblastic leukemia, chronic lymphocytic leukemia, diffuse large B-cell lymphoma, and mature B-cell malignancies were compared to determine the rates of mutation of each cancer. Immuno-oncology is a form of treatment that uses the immune system to protect against an attack on the body. There are variations to immuno-oncology such as vaccines, targeted antibodies, and various forms of gene therapy. Gene transfer is a newer form of gene therapy that can be used to treat cancer. This is done by introducing new genes to cancerous sites to begin the process of apoptosis or reduce the size of the tumor. One of the benefits to using this form of treatment include the immune system's ability to specifically target cancerous cells while leaving healthy ones intact.
Our hypothesis for this project is that while the TP53 and RAS genes, which are common mutated genes in most cancers, will also have the highest mutation frequencies in non-Hodgkin's lymphomas. Our objective is to use the CBioPortal database to analyze and compare genes with the highest mutation frequencies in certain types of non-Hodgkin's lymphomas to explore their potential in cancer therapies.
In the first step, CBioPortal was used to search for lymphoid cancer studies. Then the B lymphoblastic leukemia lymphoma studies that were selected were acute lymphoblastic leukemia 2015 and 2016 and pediatric acute lymphoid leukemia phase 2. The non-Hodgkin lymphoma studies that were selected were chronic lymphocytic leukemia, diffuse large B-cell lymphoma, and mature B-cell malignancies. In the next step, the samples from the studies were analyzed using CBioPortal to find the mutated gene frequencies, from which the 3 genes with the highest mutation frequencies in all the combined selected studies, as well as the TP53 and NRAS genes, were selected for further analysis. Lastly, the selected mutated gene frequencies were then compared to determine which had the most sample study overlap.
The study's sample analysis revealed that the genes with the highest mutation frequencies were the KMT2D, BCL2, and CRE-BBP genes, with rates of 20.5%, 14.5%, and 12% respectively. While the TP53 and RAS genes had significant rates of mutation, they only made up 9% and 3.5% of the total studied mutation frequencies. The KMT2D gene, which encodes for a histone methyltransferase, and the CRE-BBP gene, a binding protein, both act as transcriptional regulators. The BCL2 gene is an apoptosis regulator that normally blocks cell death. The analysis also yielded a comparison of the overlap between the genes of interest in the samples that were studied. The highest degree of overlap was between the CMT2D and the CRE-BBP genes, potentially indicating the presence of mutations in both genes in certain cancers.
Overall, the goal of this study was to gauge if different types of non-Hodgkin's lymphoma share similar mutated genes. Based on the results shown in Table 1 and Figure 1, the hypothesis was not supported because TP53 and NRAS did not yield the highest mutation frequencies. With the research conducted, it can be concluded that a general type of gene therapy could potentially be effective if cancers share similar mutated genes. However, if there is a substantial amount of variation between genes in certain cancers, this may not be the case. Similar to how researchers have been able to modify mRNA in vaccines to target specific proteins, they may be able to target specific genes. Furthermore, although mRNA vaccine treatments for cancer are fairly new, cancer researchers have been testing modified and unmodified forms of mRNA in their treatments. With more research on the rise, we are hopeful that scientists will be able to create effective forms of gene therapy and mRNA vaccines to treat cancer by looking at mutations that are highly frequent in most cancers. This could eventually lead to the use of gene therapies and more cancer vaccines that can be used to treat many people.